All right. So I will talk to you on metabolomic and lipidemic biomarker findings in Gulf War illness. And as advertised, my name is Gilchin Salam. Next slide.
When you're doing it.
Oh, that's right. Sorry. Trying to expand the appearance of the slides on my screen. Without much success. There we go. All right. So this is a sort of, can you see the top of the slide?
Yeah.
Okay. This is a sort of very simplified version of an oxidative stress mitochondrial model for Gulf War illness. And at the top we have exposures, and particularly organophosphate on the left and pyridostigmine bromide on the right contribute to oxidative stress, and also there's a direct line to this mitochondrial dysfunction. Oxidative stress and mitochondrial dysfunction each contribute to one another, and each actually contribute to impaired cell energy. Oxidative stress contributes to cell death by apoptosis. Impaired cell energy contributes to cell death by necrosis and sometimes apoptosis. And these together, as well as a number of other factors caused by oxidative stress and mitochondrial dysfunction, contribute to cell dysfunction, which then contributes to symptoms. And a similar version of this model, as you can look at our neural computation paper. To make it smaller so you can see. I'm having a problem. Every time I try to make it smaller to see the arrows, it makes it immediately bigger again. So I'm having trouble switching slides. Could you go ahead and do the next slide for me? Yep. So a number of Gulf War illness features are consistent with a hypothesis involving mitochondrial dysfunction and oxidative stress. Symptoms with a focus on fatigue, muscle, and brain are archetypal for mitochondrial dysfunction. So is symptom multiplicity and heterogeneity, encompassing GI, psychiatric, sleep, shortness of breath, vision, exercise intolerance, autonomic dysfunction, and the range of other problems that are reported in Gulf War illness. MD, by the way, stands for mitochondrial dysfunction, and OS for oxidative stress. Variable latency to onset of different symptoms, which is characteristic of Gulf War illness, is also classic for mitochondrial dysfunction through mitochondrial phenomena called heteroplasmy and threshold effects. Additionally, the exposure relationships observed in Gulf War illness are consistent with this hypothesis. Acetylcholinesterase inhibitors and other pesticides show a particularly strong and consistent relationship to Gulf War illness and are known to mediate some of their considerable toxicity through oxidative stress and mitochondrial dysfunction, and other chemically unrelated exposures may be contributory to common toxicity by oxidative stress. Additionally, the objective marker findings are compatible with this, with replicated documentation in Gulf War illness as reduced peroxidase, reduced heart rate variability, and other autonomic findings, reduced natural killer cell function, increases in autoantibodies, increases in inflammation and coagulation activation, which, by the way, arise from apoptosis triggered by oxidative stress, increases in gamma-glutamyl-transferase, as well as a number of other findings such as white matter abnormalities, all have a documented link to oxidative stress, and also attendant conditions that have been reported to be elevated in Gulf War veterans, such as ALS, hypertension, and the spectrum of chronic fatigue syndrome, fibromyalgia, and irritable bowel syndrome. There are a documented relationship to oxidative stress and mitochondrial dysfunction. Next slide. So this is a CDMRP, findings from a CDMRP funded effort, looking to see whether indeed there is evidence of mitochondrial dysfunction in Gulf War illness. And this uses 31-phosphorus magnetic resonance spectroscopy, which produces spectra of phosphorus-containing compounds. Phosphocreatine, or PCR, is a phosphorus-containing compound that is a backup energy source to muscle. It's depleted during exercise, and it's been shown that the recovery rate is dependent on the rate of ATP production. And therefore, phosphocreatine recovery time constant has been viewed in the literature as a robust index of mitochondrial function in vivo. And so this involves 7 veterans with Gulf War illness defined by Kansas and CDC criteria on the right, with 1-to-1 matched age, sex, ethnicity matched controls on the left. And what we see is not only is it the case that the phosphocreatine recovery time constant is prolonged in Gulf War veterans compared to their respective controls in all but one cases, but that in all but one of the controls on the left, this phosphocreatine recovery time constant is under 31 seconds, whereas in all but one of the cases on the right, the phosphocreatine recovery time constant is over 35 seconds and as high as 70. The one case that is sort of an outlier here with the phosphocreatine recovery time constant at 18 seconds, in fact, was also the one Gulf War veteran who reported their health prior to the Gulf War as not either excellent or very good, but merely as good, suggesting the possibility that this individual may have a different source of their health problem. Next slide. And so if there is mediation in part by, or in full, by mitochondrial dysfunction and oxidative stress, it might be envisaged that substances like coenzyme Q10 might confer benefit. Coenzyme Q10 is the predominant endogenous, meaning made by the body, lipophilic, meaning fat-soluble antioxidant, and it's also directly involved in respiratory chain function, that is to say, energy production by mitochondria. And there are studies showing that it minimizes oxidative stress at OS on the top, that it improves mitochondrial function in row 2, that it improves cell energy, that it reduces apoptosis, those that mediated by oxidative stress, and some forms of non-oxidative stress mediated apoptosis. And also it has improved symptoms, including some of the symptoms that have been reported by Gulf War veterans in other populations. Next slide. So based on that, we conducted the pilot study that was previously mentioned and also funded by the CDMRP to see whether coenzyme Q10 conferred benefit relative to placebo in Gulf War illness, in patients with Gulf War illness, and 46 Gulf War veterans meeting Kansas and CDC criteria were enrolled. They received 5 milligrams coenzyme Q10 versus placebo for an average of 3.5 months. And what we found was that general self-rated health, or GSRH, was significantly improved only in men. We found that male sex was a strong predictor of change in GSRH. That separately, significantly improved symptoms in the overall group, and significantly improved objectively measured function assessed by the lower extremity summary performance score in the total Gulf War veteran sample. Additionally, the improvement in function related significantly to the change in coenzyme Q10, and the improvement in GSRH related with borderline significance to the change in coenzyme Q10. Next slide. So all this is sort of background for looking at some metabolomic and lipidemic related findings. And the findings here are an effort we did with Dr. John Beepine, who was the PI, and we recruited the participants and actually did some of the analyses in this DoD-funded effort, in which he looked primarily at arachidonic acid derivatives in the form of prostaglandins, for which PG is the abbreviation, and leukotrienes, which start with L. And we see here both with logged versions and non-logged versions that particularly prostaglandin alpha, leukotriene B4, prostaglandin D2, and 13-14 dihydro-15-keto prostaglandin D2 were all lower in the case column than in the control column. And on the right, we have the mean difference, the standard error for the difference, and the p-value. So these are all strongly depressed. And again, these are all arachidonic acid derivatives. Next slide. Now, it turns out that malondialdehyde, although it is commonly thought of as a marker of oxidative stress, is also an arachidonic acid derivative. And it's a marker of oxidized fat, basically. But first, you have to have the fat in order for the fat to be oxidized. So these other findings involving arachidonic acid derivatives help to account for this unexpected finding that malondialdehyde, again in the logged or the regular version, was depressed in cases relative to controls. And on the right, again, we have the mean difference and standard error and the p-value. And below, we have the graph that shows that the case values are shifted lower. They're the red X's on the top compared to the control values, which are the blue X's on the bottom. Next slide. Now, again, malondialdehyde is an arachidonic acid derivative, as are the eicosanoids of prostaglandin and leukotrienes. And to quote from Wikipedia, malondialdehyde results from lipid peroxidation of polyunsaturated fatty acids. It's a prominent product in thromboxane A2 synthesis, wherein one of the cyclooxygenases metabolizes arachidonic acid to a prostaglandin. And I had actually asked our collaborator, John Beepine, if he was going to measure prostaglandin H2, and unfortunately that wasn't measured, so that would have been interesting to see, but probably would have also shown depression. Next slide. So this is starting to get at the issue of both oxidative stress and also lipid-related markers in Gulf-War illness. And an NIH group defined 3 candidate general oxidative stress markers, and these were malondialdehyde or MDA, F2-isoprostanes, which is abbreviated here as F2-iso, and 8-hydroxydeoxyguanosine. So malondialdehyde and F2-isoprostanes are both markers of lipid peroxidation, albeit different markers, and 8-hydroxydeoxyguanosine is a marker of oxidative damage to DNA. And what we see in a model that seeks to predict Gulf-War illness from all 3 of these is that malondialdehyde is a significant negative predictor, meaning that it's lower in veterans with Gulf-War illness, but 8-hydroxydeoxyguanosine is a significant positive predictor, meaning higher in Gulf-War veterans. So again, there is evidence of oxidative stress injury, but this does not show up with lipid-based markers, and our data suggests that this is in part because the lipids that are oxidized are depressed in Gulf War veterans. Next slide. So we also sought to see whether oxidative stress and CAC stands for citric acid cycle markers. The citric acid cycle, or Krebs cycle, is a mitochondrial cycle that is involved in energy production, predict Gulf War illness case status, and here's a model that includes malondialdehyde, which we think of here really more as a lipidomic marker rather than an oxidative marker, 8-hydroxydeoxyguanosine, which is again a marker of oxidative damage to DNA, and 2 different markers of Krebs cycle or citric acid cycle intermediate. And we see that in a model, including all 4 of these, each of them are statistically significant predictors, and we get a pseudo R squared of 0.35, which is reasonably good prediction of cases versus control status. Next slide. So by the way, that R squared reflects the fraction of the variance that is accommodated by the model. Next slide. And so here we add in prostaglandin F2 alpha in the middle, and we see that we bring the R squared in an otherwise the same model up to 0.47, and also adding in prostaglandin F2 alpha, although it is not itself significant, increases the significance of the malondialdehyde variable. So now we've gotten actually extremely good prediction of case control status, or quite strong prediction, based on a small number of lipidomic, oxidative, and mitochondrial markers. And next slide. And now we sort of can actually reduce this to 4 markers and retain the same prediction. And here we have what are really 2 lipidomic markers, malondialdehyde and prostaglandin F2 alpha, and 2 different Krebs cycle intermediates, alpha-ketoglutarate and malate. And alpha-ketoglutarate is elevated in Gulf War veterans, and in fact it has been found to be elevated in a number of conditions like autism spectrum disorder that also are known to involve oxidative stress and mitochondrial dysfunction. Next slide. So one of the things that was fascinating was that even for markers that are not themselves strongly different in Gulf War veterans and controls, the relationship to one another is different. And I'm going to call your attention first to the value at the bottom here, which is alpha-ketoglutarate and malondialdehyde. And I mentioned that alpha-ketoglutarate seems to commonly be elevated in conditions that have a known link to oxidative stress and mitochondrial dysfunction. And what we see is that in controls, it's powerfully related to malondialdehyde, which, remember, in normal conditions is considered to be a marker of oxidative stress. And in fact, the correlation coefficient is around 0.7, and there's a very powerful statistical significance demonstrated here, whereas in Gulf War illness cases, we largely lose this correlation. And what I didn't but should have shown here is that if you look at prostaglandin F2 alpha, in fact, its correlation with malondialdehyde in controls is 0.51 with a p of 0.003, but in controls, there's no relationship. Excuse me, in cases, there's no relationship. Prostaglandin F2 alpha in controls relates to alpha-ketoglutarate with a correlation coefficient of 0.72 and a p-value less than 0.0001. That is to say, it shows up on the statistical analysis as 0.0000. But in cases, it's actually a negative correlation. And similarly for gamma-glutamyl transferase, or GGT, which is an antioxidant that, like uric acid, rises adaptively in settings of oxidative stress. and in controls, it's powerfully related to prostaglandin F2 alpha with a correlation coefficient of 0.55, highly significant P of 0.001, whereas in cases, it is actually a negative correlation. So these relationships shift markedly, and the ones I showed here at the top three are actually links between citric acid cycle intermediates and markers linked to oxidative stress. Again, F2-isoprostane is a lipid marker that was considered by an NIH panel to be a candidate general marker of oxidative stress, but based on analysis using carbon tetrachloride. And so obviously it makes sense that different exposures might lead to different profiles and relationship, and that's why, of course, they called it a candidate general marker. And what these findings show is that malondialdehyde and F2-isoprostanes, which are markers of lipid, peroxidized lipids, may lose their utility as markers of oxidative stress in settings where the lipid that undergoes peroxidation is markedly depressed, like here. So what we see in these top three instances is that in controls, there's no relationship, or if anything, correlation coefficients for two of them are negative in controls, and yet powerful correlations are forged between these oxidative stress and citric acid cycle markers in Gulf War cases, suggesting coordinated modification of oxidative stress and citric acid cycle or mitochondrial variants. Next slide. And now we're going to look at a more sort of formal metabolomic analysis of Gulf War illness, and this is done using data procured from our DID study that in concert with collaborator Robert Navio here at University of California, San Diego, in concert with his group who are listed here. Next slide. And so this study, because metabolomics are different for men and women, it's important to do segregated analysis. So for this first analysis, we looked expressly at men. The sample involved 40. According to Dr. Navio, the sort of right size sample is usually 20 per group. So we have 20 male Gulf War veterans with CDC and Kansas-defined Gulf War illness, and 20 one-to-one matched male age, sex, ethnicity matched controls. Plasma was used metabolomic analysis, which was assessed by next-gen metabolomics using liquid chromatography with tandem mass spectroscopy on the machine named here, and 617 different analytes were targeted, of which 450 were detectable and 167 below the detectable limit. Next slide. So this shows a multivariate statistical ranking of these metabolites using a process called the variable importance and projection, which uses partial least squares to determine the relative importance of each of the set of predictors. And on the left, you see that a lot of these predictors are things that are called ceramides. 17 keto cholesterol is in the number 2 position. Sphingomyelin, which is SM, very different sphingomyelin. PC, which is phosphatidylcholine. So these are heavily lipid-derived markers, as well as things like valine, which is a branched chain amino acids, cytosine and guanosine, which are purines and pyrimidines. These are very strongly related. And what we see on the right, those two vertical bars, on the left are cases and on the right are controls. And red indicates that positive values of that thing strongly predict case status in the left-hand bar, and low values signify case status if it's green, and obviously the right-hand bar has the opposite profile. So all of these are extremely powerful predictors of Gulf War illness status. Next slide. So if we look at the sort of overall profile, looking at pathway names on the far left, what we see is that sphingolipid metabolism, phospholipid metabolism, cholesterol and cholesterol products, which include steroid hormones like cortisol, non-gonadal steroids, and this is the ranking produced by our collaborator. But if you look further down at item number, at vitamin D, which is the sort of 7 to 8, really number 8, vitamin D is actually also a steroid hormone that is a product of cholesterol. And number 11, bile acids are also products of cholesterol. And then we have, again, purine metabolism, and purines, by the way, are precursors for ATP. And we have eicosanoid and resolvin metabolism as number 5, and we saw eicosanoids in these prostaglandin and leukotriene phenomena. And we also have on here things like number 10, vitamin C, which again is antioxidant or oxidative stress related. Number 12, Krebs cycle, which are these intermediates involved in energy production, plasmalogens, and then as number 14, glutathione, cysteine, methionine, et cetera, which are, again, this other category of antioxidants and resulting methylation-related symptoms, cardiolipin metabolism, and then we have energy substrates involving the sugars, number 16, and the one that's off the screen is niacin, NAD, NADH, which is sort of B3 vitamin, which have a very powerful role in mitochondrial function. And next slide, please. And so to kind of review what some of these things are, for those of us that aren't very familiar, ceramides are a membrane-based lipid, and a lot of these involve membrane function, which is probably an important clue. And fortunately, we have a new CDMRP-funded study, biopsy study, in which, among other things, we hope to look at aspects of membrane composition in Gulf War veterans versus controls. And this is involved in regulating differentiation, proliferation, and apoptosis of programmed cell death of cells. Cardiolipin is actually an important component of the inner mitochondrial membrane, in fact, comprising 20% of the total lipid composition of that membrane, that is essential for optimal function of numerous enzymes involved in mitochondrial energy metabolism. And incidentally, the only known location of cardiolipin in mammals is in the inner mitochondrial membrane. The only other known location is in bacterial membranes, and of course, the belief is that mitochondria basically derived evolutionarily from bacteria. Phospholipids include phosphatidylcholine and phosphatidylinositol, which will sort of come up in a second, lipids that are major constituents of cell membranes. And plasmalogens are certain cell membrane phospholipids in which one of the fatty acids is replaced by something called an aldehyde. These are particularly prevalent in the brain, immune system, cardiovascular system, and myelin, all domains that have been reported to be affected in Gulf War veterans and are known to protect mammalian cells against damage by oxidative stress. And as mentioned, purines like adenine and guanine are not only involved in DNA and RNA, but are also part of ATP, GTP, and others of these cell energy-related compounds, and also are parents of uric acid. Next slide. So sphingomyelin, for Roberta White and Kim Sullivan, who have shown documentation of white matter abnormalities, it's typically 10 to 20 molar percent of plasma membrane lipids, and is particularly prominent in myelin. It has a role in apoptosis, in fact, by hydrolyzing into these other compounds called ceramides, and signals both when and how for cells to die. And kind of relating it to some of these other compounds, it is synthesized by transferring a phosphocholine from this phosphatidylcholine to ceramide. And I want to separately mention cortisol, which relates to, of course, to a lot of Nancy Clunis's work. She's shown alterations in hormones, including cortisol. And for those who think of cortisol as reflecting sort of a stress hormone in this setting, it's relevant to point out that it's just one of a large number of cholesterol products and steroid hormones that are altered in Gulf War illness, including bile acids, gonadal steroids, non-gonadal steroids, and vitamin D. And one can observe that cortisol alterations arguably better fit into this pattern, not of psychological stress signaling, but into a pattern of cholesterol and lipid product dysregulation. Also, we noted these Krebs cycle factors are altered relating to mitochondrial status, and also there was evidence of sugar-related or energy-related elements. And cortisol also fits into that because cortisol is also involved in raising blood glucose to assist in settings of impaired energy. You want to sort of maximize energy supply. And again, glutathione and methionine have this relationship, this important relationship as oxidative shielding or protection elements. Next slide. So these metabolomic data, and for some reason on this version I don't actually see all the points in this. I'm wondering if that means that you don't see them either. Yeah. I don't know what happened to this slide on my computer. You see the axes that show three-dimensional axes showing that Gulf War illness cases are completely discriminated from controls. And the three axes correspond to what are called latent. What happened to the slides? Latent variables or components, which are defined by partial least squares discriminant analysis, which is the standard in metabolomic analysis. So again, Gulf War illness cases completely segregated from controls using these data. Next slide. one thing that I found remarkable was the match of these metabolomic data to reported profiles of metabolomic abnormality in the setting of mitochondrial dysfunction and papers focused on the lipids of mitochondria. So the reported domains involved in metabolomic mitochondrial dysfunction are sphingolipids, ceramides, sterols, purines, vitamin D metabolism, branched chain amino acids, cardiolipin, the things that we saw in this metabolomic profile. Next slide. And the non-lipid related metabolomic markers also match, for example, the branched chain amino acids. Branched chain amino acid metabolism is performed in the mitochondrial matrix.
I'll come down right away. Thank you.
Of branched chain amino acid levels can signify mitochondrial disturbance. Next slide. And I don't have data for this. This is just what was shared with me by our collaborator, and I emailed him to try to get some data, but I have to say my mother's had some very serious health issues lately, so this kind of happened late, and I didn't get back a response. And I don't know if that's because he's not comfortable sharing his proprietary data, or if he just didn't see the email in time. But according to Dr. Navio, who has also done metabolomics and chronic fatigue syndrome and post-traumatic stress disorder, some of the metabolomic predictors were actually opposite in direction to those that he observed in his chronic fatigue syndrome group, and the metabolomic profile is quite distinct from their profile observed for post-traumatic stress disorder. He's also observed that metabolomics have often produced success in settings where genomics fail, and he cites an instance, I believe, looking at diabetes, in which high cross-geography validity was occurred with metabolomics, where highly costly efforts to find genomic replicability, even for things that were highly significant predictors at one site, failed to be reproduced at another site. And his perception, and also mine, is that our different genetics may often try to get us to the same biochemical result in response to an insult. Next slide. So these findings may have treatment implications, and also may allow us to sort of view things that we've observed through a different lens. For example, inactive 25-hydroxyvitamin D was strongly increased in Gulf veterans, which suggests a block in the renal mitochondrial 1-alpha hydroxylation that leads to production of the active 1,25-dihydroxyvitamin D form. And this is consistent with a chronic oxidative state, which is known to often provide this sort of high values of the inactive form and low values of the active form, and supports observations. Well, my sort of empirical observation of benefit in Gulf War veterans that were in my panel with cod liver oil, which is both a lipid modifying and very strongly, vitamin D containing substance. And in fact, that sort of empirical observation led me to a number of treatment trial submissions with cod liver oil. But of course, I didn't have access to these kinds of data suggesting that there were these alterations in this balance of inactive to active vitamin D at that time. And also the prominent role for phospholipids, including things like phosphatidylcholine, relates to the observation of one of our longtime veteran members of the RAC who cited material benefits from lecithin, which I believe his grandmother had recommended. And lecithin is particularly rich in phosphatidylcholine and phosphatidylinositol, and it also supports my own sort of use in my patients with mitochondria-related problems, not only of cod liver oil, but of wheat germ oil, which is the richest natural source of choline, which is important for the phosphatidylcholine. Next slide, please. So to sort of summarize some of the, quote, pre-metabolomic findings, which are also in fact metabolomic and lipidemic, but before the sort of formal metabolomic analysis, we found evidence of dysregulation affecting mitochondrial and oxidative-related parameters, and evidence of coordinated shifts in oxidative mitochondrial-related markers, leading new relationships to be forged where these were not otherwise present. We also found evidence of widespread dysregulation affecting lipid-based markers, and in the pre-metabolomic work, this focused substantially on arachidonic acid products like eicosanoids and malondialdehyde. Lipid-based markers of oxidative stress change radically in their relationship to other markers in Gulf War illness, and controls these lipid-based markers of oxidative stress relate strongly to other markers of oxidative stress, whereas in Gulf War illness cases, the other markers of oxidative stress, like 8-hydroxydeoxyguanosine or like alpha-ketoglutarate, remain high, but these lipid-based markers are depressed. Also, we found that with just a couple of markers related to mitochondrial function, these Krebs cycle intermediates, and a couple more related to either oxidative stress and or lipid alterations, we were able to separate Gulf-or-Olmos patients from controls with fairly good discrimination. But of course, it's not possible at this point to exclude factors or exposures that might have been somewhat differential in our single geographic site sample. Next slide. So to summarize the metabolomic factors, we found widespread dysregulation in a range of different lipid-related parameters, including phospholipids, sphingolipids, and that should be sterols. And these bear a strong relationship to membrane status, but also a powerful, if not absolute, resemblance to metabolomic alterations that have been shown in settings of mitochondrial dysfunction and lipid alterations shown in those settings. They also include markers that bear on oxidative stress protection, directly mitochondrial function, apoptosis or programmed cell death, and myelin production. And they provide a new lens through which to view existing objective alterations shown by others, such as corticosteroid alterations, which might signify perhaps less kind of stress alterations and more generalized cholesterol and steroid hormone product alterations. They also provide a new lens through which to consider observed or empirically observed treatment benefits, and also suggest a range of different new treatment approaches. These profiles differentiate Gulf-or-Olmos patients from other groups that have what could be considered maybe a controversial tie to Gulf-or-Olmos. There are some that consider Gulf-or-Olmos just to be no different from a generic category of chronic multi-symptom illness that would encompass CFS, but in fact, some of the markers, according to Dr. Navio, are opposite in direction to those he observes in CFS. And in fact, those who've been on this committee for a long time know that there are other studies also that show that some markers are different or opposite in direction in other chronic fatigue syndrome type cases relative to Gulf-or-Olmos. And also the markers are clearly distinct from one particular other war-related illness in the form of post-traumatic stress disorder. These metabolomic data were able to completely segregate or differentiate Gulf-or-Olmos cases from controls, and they corroborate and extend the pre-metabolic data focused on lipids, oxidative stress, and mitochondria. And we hope to replicate and extend these findings, at least we're applying to do so. And thanks very much to sort of refer to the Biorepository Consortium effort before. Dr. Sullivan and Klimas have kindly offered to participate in a proposal to replicate and extend these findings. Next slide, please. I want to thank you, and I want to have a particular few thanks here, both to the Gulf War veterans for their service and those who kindly participated in all of these studies, and also to the CDMRP. All of these data derived from CDMRP funded efforts, and I really think the CDMRP has done a really fantastic job in identifying funding. So, and I also want to thank all of you for your attention.
Thank you. Is there a pressing question that anyone has about this presentation? Nancy?
Beatrice, you alluded to some clinical trials that could follow from findings such as these. Can you elucidate just a little bit about where you think this might take us from the perspective of treatment?
Well, obviously it both supports some of the existing things like the studies that are going to sort of mitochondrial directions, but also membrane-based therapy would be an example of many other potential studies that these data would help to define.
Thank you very much. So we will now move on to the next three presentations. I think this will be the new opportunity to shine. And two, they always shine. Except at the Meadowlands.
Oh, they never shine.
So the first two will be this morning, and then the third after lunch. And all three are at the East Orange VA healthcare system. and the War-Related Illness and Injury Study Center, which is one of 3 nationally. Our first presenter is Dr. Falvo. Michael Falvo is a PhD scientist, a clinical exercise physiologist at the New Jersey Center. Dr. Falvo's research interests include the study of physical and environmental stress on cardiopulmonary, autonomic, and bioenergetic function. In his clinical role, Dr. Falvo supervises cardiopulmonary exercise testing and pulmonary function testing in veterans with post-deployment health issues. Dr. Falvo, thank you for joining us today.
Thank you, Dr. Howard, and thank you for the invitation. We appreciate being here today. So this is probably very appropriate to follow Dr. Bowen's work as well, because we're going to stick with this mitochondria theme. Just get oriented over here and see if I can expand this for you guys. So first, before I start, I want to say the views today on my own and do not necessarily reflect the opinions of the VA government. And so here's our beautiful image of a scanning electron microscope image of mitochondria. This is in the human world. So typically when we look at our biology textbooks, we see mitochondria look like kidney bean shaped types of things. This is a cross section of those. And inside of those is kind of where the magic happens. We'll talk a little bit about that. But before I do, let's talk about how many mitochondria we have. And this number really sounds fake. It sounds like a little kid describing how much money or wishes they have. So 10 million billion is what's put out there as an estimate of how many mitochondria we have in our body. That accounts for an estimate of approximately 10% of our body weight. The interesting thing about mitochondria is that the more metabolism that goes on in those specific cells, the more mitochondria there are. So in other words, brain cells, skeleton, skeletal muscle, they're going to have a larger percentage of mitochondria per cell than other organ systems. Now, in biology, we heard that mitochondria are a powerhouse. This is where ATP is generated, and this is where energy occurs, and that's all true. But we've learned a lot over the past decades and years over what else the mitochondria is involved in, and that includes some things that Dr. Goldman mentioned, apoptosis, calcium homeostasis, as well as heme and iron sulfur cluster assembly. And I'll touch on those in a little bit. So importantly, we're going to talk today a little bit about mitochondrial DNA. And only in the early 1960s do we even recognize that mitochondria have their own DNA. They have about 2 to 10 copies of their DNA contained within the mitochondria. And we refer to that as mitochondrial DNA copy number. So the words in bold right now are the things that I'll come back to later, which is why I want to introduce them. So mitochondrial DNA encodes proteins, specifically 13 proteins, for respiratory chain complexes, also known as the electron transport chain, critically important in generation of ATP. Now, when we talk about mitochondrial dysfunction, you can see kind of where that starts or where that comes from. And Dr. Letty made a good point earlier about kind of digging more into some of the mechanisms involved with how these things present themselves. So we're particularly interested in whether mitochondrial DNA was damaged. Does that contribute or lead to mitochondrial dysfunction? Now, what's always been interesting to me when I first joined the VA back in 2010 is that the symptom overlap of Gulf War illness and that of what the NIH refers to as mitochondrial disorders appears very similar. So the National Institutes of Health defines mitochondrial disorders as a clinically heterogeneous group of disorders. It suggests that many involve multiple organ systems and that often they present with prominent neurologic and myopathic features. So to me, that always made sense to me as a scientist. So Dr. Goldman presented in a great 2012 paper this hypothesis of mitochondrial dysfunction in Gulf War illness. And so what I wanted to do is just describe that briefly as it pertains to our specific variables of interest in this study. So if we assume that there is some type of trigger, whether that's an exposure, a mix of exposures that could generate reactive oxygen species, or what leads to oxidative stress, that could tip the balance in the body. In other words, the amount of oxidants outweigh the body's ability to defend itself in the form of antioxidant capabilities. So the body's first kind of acute reaction to that, or at least the mitochondria's first reaction to that, is to make more of itself. And so to account for damaged mitochondria in the face of oxidative stress, it can make more of itself, and we refer to that as mitochondrial DNA content, and that's something we can measure. That's an acute scenario, but chronically over time, if there's persistent inflammation, persistent oxidative stress, that can contribute and then lead to mitochondrial DNA damage. Once the mitochondrial DNA is damaged, that could further lead to increased reactive oxygen species, creating a vicious cycle, which then kind of furthers that illness, but also could lead to increased mitochondrial dysfunction. So here's a busy slide. I'm just going to walk you through just a couple points here. So I stole this slide from my colleague, Joel Myers, down at Duke. He's a mitochondrial toxicologist. He's helped us a lot with this project and some future work. So he talks about why mitochondria specifically are they particularly susceptible to injury. And so he put this great paper out in 2013 and talked about environmental targets, environmental toxicants targeting mitochondria. Because mitochondria is particularly susceptible and vulnerable to injury. A couple key points about the mitochondria is that its DNA, in comparison to nuclear DNA, is less efficient in repairing itself. It lacks certain things that protect itself from insults. So that's one thing that makes mitochondrial DNA more susceptible to damage. Dr. Golem mentioned the rich phospholipid bilayer. That is particularly susceptible to injury to certain environmental toxicants. We even know that certain metals can actually accumulate into the mitochondria. So it seems that, indeed, environmental toxicants can specifically target the mitochondria. And this is a very rapidly growing field. So what I wanted to do now is kind of talk a little bit about a study we're completing right now. The study was supported by the VA Office of Research Development. It was a pilot award, so a 2-year project. And so in this study, we recruited over 50 individuals to participate, 30 of whom met criteria for case definition and Gulf War using Kansas Steel criteria. So we brought them in for 2 days. They participated in multiple bouts of exercise. And then we also drew blood samples to look at some of these mitochondrial markers. So I'm going to talk about this in 2 parts. The first part I'll talk about is our blood samples here.
Could you tell us what the controls are?
Sure, the controls were similar age, gender, not meeting criteria for Gulf War illness.
Were they veterans or were they?
The majority of them were veterans. We have included a couple civilians. Those are not presented here today because we were trying to wrap up the study, so there's a couple. So this is very preliminary. We're still trying to recruit the additional group. And so we did use the Kansas Steel criteria to identify cases or controls. No, thank you for clarifying. So this preliminary data we'll present right now is just the analysis of the blood samples. And so what we have now is a complete analysis on 25 Gulf veterans with Gulf War illness, and we're able to complete analysis on 6 controls. So to tell you a little bit about the analysis we did, so again, it started from blood. So we took a blood sample, we centrifuged that blood sample, we isolated out through a procedure just the white blood cells. And the white blood cells are important because that's where we contain mitochondria. Red blood cells are devoid of mitochondria. So after we're able to isolate out those white blood cells, we can extract total DNA from those white blood cells. Once we extract total DNA, we can perform an experiment that tries to identify a few different things. And I'm going to just kind of keep it relatively simple here. So the first one is mitochondrial DNA content, which we're going to call copy number. The second thing is actually looking at mitochondrial DNA damage, which we'll refer to as lesions. So is the mitochondrial genome damaged? Also, we looked at the nuclear DNA genome and see if there was lesions there as well. So those are the 3 primary things we're looking at. So the first, before I get into the results of each 3 of those, I want to just touch on briefly how we are operationally defining damage here. So this quantitative PCR is an assay developed by some folks that we work with, Dr. Janine Santos and Dr. Joel Meyer, that performs this amplification of mitochondrial DNA so that when damage is present, sorry about that. So when no damage is present, the way this kind of technique works is that you can amplify long segments of the DNA. However, if there's damage or lesions that are present, it won't be able to amplify the entire longer piece, that longer amplicon of the DNA. So what we do is that we can then make that, we can look at a longer piece, and then we can look at a smaller piece of the mitochondrial DNA, and we look at that ratio, and that ratio gives us some evidence of damage. I had 3. So Dr. Kalasinski, is the slides on here? So we have that's the meat of my study here, so the 2 graphs that are missing.
We got them in our book. We got them in our book.
Okay. Is it present on? I could talk through it, I apologize.
You can just keep talking.
Okay.
Yeah, we got people on the line that need to see this stuff.
So I'll go off these real quick and then I'll go back. Yeah, this is the important one. So we'll start first with mitochondrial DNA copy number. So again, this is the content. And we see that veterans with global illness have a significantly more copy number, about 50% more than our control group, and 22 veterans relative to 6 controls. And so this slide talks about the DNA damage, both mitochondrial and nuclear. So on the left side of the screen, we have veterans with global illness. In the blue, we have mitochondrial DNA damage. And in the red, we have nuclear DNA damage. And the controls are on the right side of the screen here. So what we see is a significantly larger amount of DNA lesions, both in the mitochondrial genome as well as the nuclear genome. But importantly, this kind of supports some prior literature that, as you can see, the mitochondrial DNA seems to have significantly more lesions than the nuclear DNA. And that is supportive of prior research as well. So let me jump back to the Adobe Connect. Okay. Sorry for posting the phone. So we established, well not established, so this our preliminary data support that copy number and nuclear DNA damage and mitochondrial DNA damage are higher in veterans with global illness. We also wanted to look a little bit about mitochondrial function. And so one thing again about mitochondrial DNA is that it encodes proteins for the respiratory chain complexes. As you can see here, complexes 1 through 4. Again, this is, if you can think of the complexes as producing ATP, and the complexes produce ATP. And if you think of ATP as money, the complexes are kind of like the mint. They're producing the energy, if you will. And sorry about that, folks. Let me jump back. So here is the complex 1 activity. So what we see here on the bottom is mitochondrial DNA lesions. And on the y-axis, or the vertical axis, we see complex 1 enzyme activity. And we see relationships such that the more DNA lesions that are present in mitochondrial DNA, the less enzyme activity for complex 1. And that also is consistent with complex 4. But I do have to mention this is a smaller sample, not the whole sample we've completed yet. Those analyses are still underway. So, like complex 1, complex 4 also demonstrating that the more mitochondrial DNA lesions that are present, the less enzyme activity. And we also did look at a plasma reactive oxygen species analysis, just to, as Dr. Bowen mentioned, oxidative stress is present. Obviously, that's related to mitochondria both generates and is damaged by oxidative stress. We see a non-significant, albeit a trend, for mitochondrial DNA lesions, such that the more lesions, the increased amount of reactive oxygen species. I'm going to follow up that with some brief exercise data. So this is completed data we have right now on 22 global veterans with global illness. There's control, I'll present that in a minute. So to step back just briefly on kind of why we're interested in the exercise data as well. So as an exercise physiologist, I'm really interested in the coupling of internal and external respiration. So if we think of this graph right here as kind of the exchange of gases both from the environment to the blood, then to the cells, and then vice versa, from mitochondria down here, through the blood, and out through the mouth. Those are things potentially we can measure. So one specific thing that we're interested in measuring is what's called the ventatory threshold. This is also in terms of the lactate threshold or anaerobic threshold. And essentially what it provides is the point or that deflection in which we shift from aerobic metabolism to anaerobic metabolism. That point is represented by this dashed line right here in red. And what this indicates is that in the beginning portion of an exercise test, if anyone's ever done an incremental exercise test, think of this as kind of riding a bike up a hill. In the early portion of that activity, ATP is generated from mitochondrial respiration. But at a point at which that intensity starts to increase and it's challenging to keep up, we start to shift over to anaerobic metabolism. And that shift tells us a lot of good information. We know that in healthy, even sedentary people, that shift occurs around 50 to 65% of peak exercise. What we think, and what's been demonstrated in patients that have mitochondrial disorders, And there's other factors that can contribute to this, but it's an earlier onset of that inventory threshold. So that's one of the things that we're particularly interested in. So here are some demographics from that sample. Again, these are the same folks that we looked at the mitochondrial DNA lesions from as well. Right now we've completed 24 veterans with global illness, 24 completed, 22 controls. Similar age, similar body mass index, similar self-reported exercise or physical activity per week. No change in smoking habits. Fatigue severity is reported here. This just makes sense with global illness. The fatigue severity score is based on the fatigue severity scale, where a score of over 35 indicates clinical fatigue, and then deployment length at the bottom. So those two things were different between groups. So what we saw in this preliminary analysis is that the veterans with global illness and the controls both achieved a similar exercise capacity. In other words, we saw no difference between the maximum level of exercise they were able to achieve. And I'm going to have to apologize, and I hate doing that, but I made an error on the slide. I switched them. And so what this is talking about here is the time compensatory threshold. So on the left side here it says global illness. I wrote that incorrectly, that is controls. So what we did find is that veterans with global illness had an earlier onset of the compensatory threshold in comparison to controls. And if we take that back to the compensatory threshold concept here, I think that's supportive of this relationship with mitochondrial dysfunction. To look at that a little bit further, in the veterans with global illness, we looked at mitochondrial DNA lesions and its relationship to the time at which the compensatory threshold occurred. And what we found is that the more mitochondrial DNA lesions the individual had, the earlier onset of the compensatory threshold. So to wrap up some preliminary findings, what we've seen so far in our data is that we observed an increased mitochondrial DNA copy number, increased mitochondrial DNA and nuclear DNA damage in veterans with global illness. We found that mitochondrial DNA damage is greater than the nuclear DNA damage that was present. We found that mitochondrial DNA lesions were associated with complex 1 and complex 4 activity. We also found that lesions were associated with an earlier onset of the compensatory threshold. So I said right now we are literally probably just wrapped up the last few subjects in the prior weeks, and we're completing the analysis and finalizing that. We're going to give the committee and the folks in the room kind of a peek about what we're doing next. So, like Dr. Blighty mentioned, we think it's important to dig down into the mechanisms. So this was a VA-supported pilot study. So where we want to go from here is better understand mitochondrial DNA repair pathways and bioenergetic mechanisms. So in order to do that, you need to go find smart people. And so I found a really smart collaborator down at Duke, who's a mitochondrial toxicologist, and we're submitting a CMRP proposal in a couple weeks. One interesting thing is that we kind of happened to stumble on is that there's a collaborator at our institution that is a red blood cell expert, and we talked about this idea of mitochondria influencing red blood cell production and function. Importantly, because heme synthesis takes place in mitochondria. So we've got a VA pilot proposal that's currently underway. And then we're really excited right now for a new proposal that's just awarded, which we're just getting started right now. That is a collaborative effort both with us here in New Jersey and Dr. Dane Cook out of the Madison, Wisconsin VA. And so this proposal is supported by the VAs called Post-Exertion Malaise and Global Illness, Brain, Autonomic, and Behavioral Interactions. So again, we teamed up some really smart people, like Dr. Dane Cook and Dr. Glenn Wiley, you're going to hear from next. And with that, I'd like to thank everyone's attention. Thank you for coming. Thank you for the opportunity to speak today. I also wanted to acknowledge some fantastic people that work in my lab. The VA Office of Research and Development for supporting this pilot project, the New Jersey RISC, the veterans that took place and participated in our study, and some really fantastic collaborators. I'd be happy to take any questions.
So maybe I'll ask a question about controls, which is always the critical question in human research. And your GWI group were heavier smokers, and that must affect VT.
VT, perhaps. It depends on kind of the length of smoking. So I guess your follow-up question probably would be about mitochondrial DNA lesions as well. So we did examine self-reported hackers and its relationship and found them to mitochondrial DNA lesions as well as exercise performance. We are running some analysis right now to look at continuity to match that up as well, to make sure that that's not kind of accounting for the majority of the variance. That's a critical piece. Smoking, physical activity, lots of other dietary habits could potentially influence this. Yes, sir.
2 questions. One is, have you thought about doing muscle biopsies and looking at things there, given the issues of heteroplasmy and differences between circulating mitochondrial DNA and muscle?
Absolutely. We have thought about that. You're 100% correct that the skeletal muscle provides a much richer opportunity to examine mitochondrial function, both in terms of intensity and function perspective. So we went with red blood cells for 2 reasons. So one, for a pilot study to see if we can establish before we move to something more invasive. And then the second thing is, what we've been concerned about is that the symptoms present much more than just in the skeletal muscle. And, we obviously can't take brain tissue, so it's another way for us to look at maybe something that's circulating or more representative of other organ systems. So it's a balance, you're right. I think importantly what we wanted to do here was to establish and potentially move to more metabolically active tissue.
Part 2, and you may know the answer, how do these changes relate to changes, if any, that are seen in POTS?
That's a great question. I don't know the answer to that. I know we didn't have any folks that had orthostatic hypertension in this that participated in this study. That's an interesting question. I don't know. Thank you.
Can I make a comment?
Please.
I just wanted to mention that we are initiating a muscle biopsy study, and one relative advantage of muscle, even if what you're interested is in the brain, is that it is a post-mitotic, highly energy-demanding tissue that can relatively accumulate DNA injury. And so for that reason, sort of post-mitotic tissues like muscle and brain are thought to perhaps document or demonstrate mitochondrial injury more than tissues that overturn regularly, like circulating cells.
Thank you.
Excellent. Thank you so much.
Thank you very much.
Our next speaker, also from the East Orange VA healthcare system, is Dr. Glenn Wiley. Dr. Wiley is Associate Director both for the Neuroimaging Center and Neuropsychology, and the neuroscience research at the Kessler Foundation, and is also an associate professor at Rutgers, New Jersey Medical School. His research interests fall into 3 broad categories: cognitive control, cognitive fatigue, and the neurophysiologic effects of cognitive interventions. Dr. Wiley, thank you very much for joining us.
Well, thank you. Let's see. All right. Can everyone hear me? Mm-hmm. Yeah, good, okay. This talk is sort of a change of gears, right? Because we've been looking at this microscopic stuff. This is going to be macroscopic. And I'm going to be talking about fatigue and Gulf War illness using functional neuroimaging, which is why I'm showing all these interesting pictures. All right. All right. Same disclaimer as Mike gave. These views are my views. All right. And to start off, who's affected by fatigue? We are in DC. Democrats are affected by fatigue. And Republicans are affected by fatigue. Everyone is affected by fatigue. And to come back to Gulf War illness and fatigue, we know that 25 to 35% of Gulf War veterans report chronic multi-symptom illness, which is characterized by fatigue, pain, and cognitive complaints. And that's a lot of people, up to 250,000 people. And the prevalence of fatigue in this population is pretty high. So at the risk, out of 224 Gulf vets that we saw, 64 reported severe pain and fatigue. And those were not just people with Gulf War illness, those were Gulf War vets. And in an online survey of 542 Gulf vets with Gulf War illness, 94% reported significant fatigue, so that's a big problem. And then I thought I'd do a little bit of background about what fatigue is, because it's sort of this amorphous concept. And I gave some background, which I cut out last night, but then we couldn't update the slides. So the background is in the next couple slides, which I'll skip over, but I think this is important. So what does fatigue feel like? And this is from a guy who actually had TBI, not Gulf War illness, but it makes a point that I wanted to make. So he said, the more distractions there are, the more cognitive energy that must be expended to filter out background noise, increasing the attentional demands of my brain to complete each task. Everything takes more cognitive energy after a serious brain injury, and cognitive energy is what I wanted to sort of focus on here, because that'll be important as we go along. So, because we're talking about cognitive fatigue, so I'll come back to that. Okay, this is what fatigue is, some background. I'm not going to go into that, or that, or that. I can come back to these things. Okay, what I wanted to come to was types of fatigue. So there's physical or motor fatigue. So this is muscle weakness, or the ability to maintain motor output. This is like if you run a marathon, when you're done running a marathon, your muscles are tired, right? That's muscle fatigue. There's also motor fatigue. This is more central, so this is a loss of maximal capacity to generate force during exercise. This is more like Mike stuff. And so this is like this is the motor strip telling your muscles to work, and you lose the ability to do that over time. But I'm not talking about either of these today, okay? So there is physical or motor fatigue, but that's not what I'm going to talk about. I'm going to talk about cognitive fatigue. So cognitive fatigue is feeling mentally drained after doing cognitive work, and it's a perceived difficulty in maintaining attention. And it's been studied in two ways. It's been studied over a period of time. So, for example, during the course of the workday, you get up in the morning, you're rearing to go. By the end of the day, you're tired, you want to go to bed. That's one way that fatigue has been looked at, cognitive fatigue has been looked at. Another way is during sustained mental effort. So, for example, assessing fatigue before and after the performance of a demanding task. And that's what I'm going to talk about today. All right, so how do we study fatigue? And for those of you in Boston, I thought I'd use shoveling snow as an example.
Oh, thanks for reminding us.
Yeah.
It's coming soon.
That's right. Hopefully not as bad this year. Okay, so let's think about shoveling snow for a minute. And if you have shoveling performance here on the y-axis, and you have 3 epochs of shoveling on the x-axis, you might imagine going out and shoveling snow, and you go out, and when you start, you're near the top of your game, right? You can shovel like anything. And but after a little while, your shoveling performance declines. You start to get tired. You go in, you get a cup of coffee, you come back out, and you start again. And your shoveling performance is a little bit better than when you went in. Maybe not as good as when you first started, but and your performance falls off even faster. You go in, you get warmed up, you come out again, same thing. Your shoveling performance is a little bit better than when you went in, but it falls off even faster. And you better be done now because you're almost at the bottom of shoveling performance. So that's a pretty good model for physical fatigue, right? Okay, so let's turn to cognitive fatigue. And let's just change the axis. So we have a cognitively demanding task of some sort. We have performance on the y-axis, and you have 3 epochs. And if you assess fatigue before and after each epoch by asking people how mentally fatigued they feel, you might find that as performance falls off, their fatigue goes up, right? It sort of makes sense. And this is how people have been trying to look at fatigue for over 100 years. But this is a setup. Even though it's really sort of intuitive and you all followed me up to this point, in fact, in over 100 years of research, it's been shown that there is no relationship between subjective feelings of fatigue and objective measures of performance, like shoveling, or, or reaction time performance on some cognitive task. Even though, from a scientific standpoint, it would be nice if performance suffered as fatigue increased. And actually, that was something Mike just showed. That in the exercise stuff, they were able. These guys who we know are fatigued were able to perform just as well as the control. So it's been a problem. It's been a problem for over 100 years in the study of fatigue. So that's why we started to look at the brain and to use neuroimaging measures to look at fatigue. So if subjective reports and objective measures of performance don't correlate, but we believe that all psychological states are based on activity in the brain. We are materialists, after all. And so even subjective reports of fatigue should have some sort of neural correlate. And so perhaps brain activity can be used as an objective measure of fatigue. And indeed, in 2000, Chiduri and Bahan came up with this model of fatigue. They based it on Parkinson's disease, where fatigue is, a big problem. It's one of the ways you diagnose Parkinson's disease. And so if we cut open the brain, they said, okay, so the basal ganglia is important for fatigue because Parkinson's disease attacks the basal ganglia. And I'm just I painted everything in the basal ganglia here, and other areas too. The network is a little bit more discrete than what I'm showing here, but this gets you the idea. The thalamus is also important for fatigue. Frontal cortex, there I painted all of frontal cortex, but certain areas of frontal cortex are important for fatigue. Anterior cingulate and ventromedial prefrontal cortex are important for fatigue. And yeah, and that was their model. Turns out the parietal cortex is also important, but we'll come to that. So that's the background I wanted to give you, and now I want to talk about the study that we've been working on and is currently ongoing. And I should have said at the beginning, ask questions as we go along if you have them. Okay. So we used a working memory paradigm to induce fatigue in 21 veterans with Gulf War illness, and 9 matched health control veterans, also from the same era, before anyone asks. And we acquired fMRI while they performed 4 blocks of the 0 back task. I'll come on to what that is in just a minute, that task is less fatiguing, and they performed 4 blocks of the 2 back task, which is more fatiguing. I'll explain that too. And the order in which those were administered was counterbalanced across people. And we got subjective reports of fatigue before and after each block. So here's the paradigm we used to elicit fatigue. It was the N-back task. That's what it's called, and it's a matching task. And we use 2 levels. The 0 back task, you match each item against an item that you hold in memory. So before they start, we say to the subject, respond when you see a K. And so they see these letters on the screen one at a time. See an N, it goes away. You see an R, it goes away. You see an S, it goes away. They see a K, they respond. That's a 0 back task. It's basically a visual matching task. It's pretty easy. The 2 back task is much more challenging. And in this task, you match each item against an item 2 back in the list. And so you see an R, it goes away. You see an N, you see an R, you see a Q, you see a K, you see another Q, and there you should respond because it matches the Q 2 back in the list. So you have to hold items online in memory. You have to remember that Q 2 back in the list, even though you're also remembering K because it might be relevant later, and the 2 back task is very challenging. Okay. We asked about fatigue before and after each of these blocks. But we didn't want to be too transparent about what we were doing. So we also asked about happiness, sadness, anger, pain, and tension. And those were all randomly varied in terms of the order we asked those in every time we asked them. So here is sort of method. So this is an example of someone who got the 0 back task first for 4 blocks, and then the 2 back task. And before and after each block, we asked, how mentally fatigued do you feel right now at this moment? And they rated it on a scale of 0 to 100. And before they went in, we told them that we told them not to be consistent. Consistency was not what we were after. So if they said that they were fatigued 10 on one, on one block, before one block, they could go up or down. So we just wanted to know what they were feeling right there, instantaneously, at that moment. It didn't matter what they felt before. So we tried, All right. Here's our sample. 21 folks with Gulf War illness, 21 controls. Sorry, 9 controls. They're matched in age. They're matched in education. They're matched in gender distribution. Almost all of them were males. All right. What's this? Why am I back to the beginning? That's interesting.
This is one of those bar graph ones.
Yeah. Okay. So wait, so if I go over here. Desktop. Go back. Go up. Okay. What's that for? All right. Yeah, the fatigue's okay. It's the task switching that's troublesome. Yeah. All right. All right. So, what's that good? Come on. Hey. All right. So here's their behavior. This is their response time, and what I'm plotting here is response time on the y-axis, the 4 blocks on the x-axis, the zero back task in blue, and the 2 back task in orange. And you can see there's a main effective task such that everyone responded slower on the 2 back task. The 0 back task, it's a harder task. And there was an interaction of task and block. And that's because the 0 back task, they actually responded more slowly on as they as they went along. Really, and that's really block 3, which you can see. I'm not sure what that's about, and I'm not sure that that's going to stay as we have more subjects. But yeah, that's what that's about. But the main point I wanted to make with this slide is that there's no interaction with group, or a main effective group. In response time, and error rates, which I'm not showing you here, are the same as true. There was an effective task and nothing else in the error rates. So, once again, even though these guys are more fatigued, they, which I'll show you in just a minute, the individuals with Gulf War illness were more fatigued. Their behavior was just as good as the controls. Okay, so here are the fatigue ratings. And these are the ratings that we collected before and after every block as they were going along. And so this is the visual analog scale of fatigue, which is that 0 to 100 scale I showed you. Time along the bottom are the 5 observations, because this is before and after each of 4 blocks, so you have 5 observations. And there was a main effective group, so you can see that the folks with Gulf War illness, they're in red, had more fatigue than the veterans with not with Gulf War illness. And there was a main effective observation such that everyone showed more fatigue across time. There was almost an interaction of group and observation, which I'm thinking will probably come up out of the noise because it really looks like it ought to be there in my graph. So I think it will come up out of the noise as we get more subjects. Okay. And in terms of neuroimaging findings, we found fatigue-related activation in 4 areas. One of them is this ventromedial, prefrontal cortex, which sort of started on the ventral surface and extended laterally. And so I'm kind of, in this slide, I sort of picked a hot spot that was kind of in the middle of that cluster. And so these are the 3 views, for those of you who aren't used to looking at brains. This is the axial view, so the brain cut this way. This is the coronal view, so the brain cut this way. And this is the sagittal view, so the brain cut this way. And then this is a 3D rendering of the same thing, just to kind of give you a better idea of where it is in space.
Can you tell us what the timing was of this in relationship to yes.
The timing. So we acquired the fMRI data during task performance. So this is sort of data from that whole, all 8 blocks.
I see.
Yep. And that's kind of what I'm showing you here. So the over here, this graph is the interaction of fatigue and percent signal change in the ventromedial prefrontal cortex. And so on this, so that's, the graph is still there. And so what you can see here is that for folks with Gulf War illness, shown in red, as activity increased in ventromedial prefrontal cortex, fatigue decreased. And there was a reliable correlation, negative correlation of -0.42. Whereas for the controls, as activity increased, fatigue also increased. So
just for my own, maybe I didn't follow, there are 9 controls and 21 individuals, so they're being measured multiple times? Is that why there are
So, yeah,
many more circles?
oh, yes, absolutely, yeah. So, yeah, because we measured, so I didn't actually tell you what the model was. So the way we analyzed it was we did an ANCOVA, and we had the activation for each of the 4 blocks for each of the 2 tasks, so 8 blocks. And in that we put the fatigue rating, we put run to model the fact that, or observation to model the fact that we had 4 runs for each task. And we put task. And so this plot shows, is all that data. So there are a lot more than 9 points, blue points, because we have 8 points for each person.
And it looks as if the controls are at the 2 edges of the fatigue, improved and worse, and the patients are in the middle, is that?
Well, the patients, I think, so I see what you mean by the bimodal distribution of the controls. The patients are kind of, they're all sort of negative, or they're clustered more to the negative side than the positive side, but yeah.
So why are the controls so bimodal?
What's that?
Why are the controls so
I don't know,
bimodal?
and I don't know if that'll hold up. These are 9 people, so.
And are they segregating with individuals? Is the same?
That is a level of detail I haven't looked at. It's a good question. It's a good thing to look at, for sure. Yep. So this pattern was true in all 4 areas where we found fatigue-related activation, where we found fatigue interact with group. So here in the cingulate, we find again the same pattern in terms of the correlation, but this is the cingulate, so it's kind of in the middle here, down between the 2 hemispheres. These 3 pictures of the same orthogonal views, and this is the 3D reconstruction again. And then again, for Gulf War illness, for folks with Gulf War illness, as activity increased, fatigue decreased, a less strong correlation, but nevertheless reliable. And for the controls, as activity increased, fatigue also increased. Again, a less strong correlation. And again, for the controls, a bimodal distribution. Back here in lingual gyrus, we found the same pattern. So this is the visual area, probably devoted to decoding those letters we were showing them. And again, the 3 views here and 3D reconstruction. And in terms of the interaction, again, for folks with Gulf War illness, as activity increased, fatigue decreased. And for controls, as activity increased, fatigue also increased. And then finally in the precuneus, which is the parietal area, so up here, we found the same pattern. So these are again the 3 orthogonal views and the 3D reconstruction. And for veterans with Gulf War illness, activity increased, fatigue decreased. And for controls, as activity increased, fatigue increased. So, okay, so what does all this mean? So the first point to make is that we're able to measure changes in the brain associated with fatigue. So it looks like we actually have a way to objectively measure fatigue in this population, and we've done this same sort of thing in folks with TBI and folks with MS as well, and we're able to find fatigue-related activation in those folks too, in similar areas. So that's great, right? Because we have something objective we can actually measure, and performance is not like that. Okay, so that's a good thing. Something worth crowing about a little bit. We find fatigue-related activation changes in areas implicated in other populations, such as MS, TBI, like I just said, such as ventromedial prefrontal cortex and parietal areas. So we find, back to the Chidori-Bahan model, the basal ganglia is heavily interconnected with ventromedial prefrontal cortex. There's a reward circuitry there, such that, yeah, when you get a reward, the ventromedial prefrontal cortex gets activated, and the caudate of the striatum also carries about reward, and these are areas we find related to fatigue. So, and in all cases, as activity increased, fatigue decreased in Gulf War illness. And so this suggests, at least to me, that this activation is combating fatigue. Because you can imagine two possible interpretations, right? This is fatigue-related activation, that's how I've sort of been talking about it. But you don't know why, we don't know why. It's there. We don't know what it means. It could mean that, that these are areas that are always active when people are fatigued because it is somehow the brain's instantiation of fatigue, whatever that would mean. And when you feel fatigued, these areas are active. It could mean that these folks are getting fatigued and they're pushing through and they're getting the job done, and these areas are active because they're fighting off fatigue. And I think that's what this negative correlation means, because the more activation there is, the less fatigue these guys are feeling. So in order to do that, in order to really sort of nail it down, you need to do longitudinal studies, and we're working on that. So here's a speculative interpretation, which is a dopamine imbalance hypothesis. And we came up with this on the strength of other data in TBI and in MS, but it seems to fit these data pretty well. And the idea here is that there's a sweet spot for dopamine in your brain, and if you have too little dopamine, you feel fatigue. If you have too much dopamine, you feel fatigue. And so if you're down here, you have damage to your brain, or you have too little dopamine, then as you get more activation in the dopaminergic areas, like ventromedial prefrontal cortex, you'll move along here and you'll have less fatigue. Whereas if you're already pretty much there, like healthy controls presumably, if they get more activation in those dopaminergic areas, they're going to move up here and they're going to actually feel more fatigue. And so this may be why, this is highly speculative, and based on a relatively small sample. Nevertheless, this may be why we find that interaction where the healthy controls are showing more fatigue as they get more activation, so that they're going up this slope, whereas the folks with Gulf War illness are showing less fatigue as they get more activation. Okay. Oh, right, future directions. Oh, one thing I was also planning to say, which I forgot, was that some of these Gulf War illness, some of these subjects, from both groups, I think, also participated in Mike's study. So there was some cross-pollination there. But in terms of future directions, we have a plan to submit a CDMRP grant, or we have been asked to submit a CDMRP grant to use a home-based, individually calibrated exercise intervention to alleviate fatigue in Gulf War illness. So this is something that Mike and I are going to do in the coming weeks. And then we're also interested in the interaction of physical and cognitive fatigue in Gulf War illness. So up till now, Mike's been putting people on his exercise bike in his lab, and I've been putting people into my scanner. And now we want to do those two things sort of, right after the other, one right after the other. So look at how being physically fatigued affects mental fatigue, and how being mentally fatigued affects physical performance. And that's pretty much all I want to say. I need to give thanks to all these good people who helped, and to the veterans who participated, and to the VA who gave me, who gave me the grant. That was it. And that's it.
Thank you, Dr.
My question kind of relates to two things. One is semantics, and the other is the degree of severity of a condition. So your wording is as activation increases, fatigue does something.
Right.
And when I think of the use of fMRI in clinical research and exposure-related research, where I've done it a lot, I think of it the other way. So as exposure increases, something happens to activation.
Okay.
So, I mean, I don't know if you say it that way because it's a biomarker, it's just a different way of thinking about it, but it relates to where I go from there, because with some conditions, like say early Huntington's disease, early Parkinson's disease, chemical exposures that are sufficient to cause some measurable changes in cognition but don't cause dementia or, global cognitive impairment, you get this hyperactivation in the mild stages, and then when it gets very severe, it's the opposite. So I'm wondering if you have patients who vary in terms of, Gulf War illness patients, who vary in terms of the severity of their illness and their susceptibility to cognitive fatigue, if you might see something bimodal, particularly if you increase the difficulty level of the challenge.
Yeah.
Just an idea.
Yeah, and I think it's something to look at. There certainly is variance in the fatigue ratings that we get from these guys, from both the controls and the folks with Gulf War illness. I just know that anecdotally, and as we get a bigger sample, we'll be able to sort of break it into groups.
That would affect how your biomarker works.
Yeah, and one thing that we had expected, and when we wrote the grant, we wrote it this way, was that we were going to be able to induce fatigue. The 2-back task, which is hard, is going to induce a lot more fatigue than the 0-back task, which is easy. That's not how it works. It's time on task, and which is really surprising to me. But these guys, they just get more fatigued as the task goes along, and the difficulty of the task doesn't have as big an effect as I expected, and it doesn't have a big enough effect to come up out of the noise in the data.
Yeah.
Maybe it's there, but
Another thing is with CPT, sometimes you see that pattern where there's in normals where there's increasing fatigue or slowing, and then they speed up again, like you saw,
Yep.
that block in the middle.
oh right, yeah.
So I've
Yeah.
seen that over and over and over.
Yeah.
So I don't know if it's going to go away with more subjects or not, but
We'll
but
see.
there's precedent.
Okay, that's good to know. Thank you. When I write this up. Yeah.
So I think it was also from your site, you moved your mind's earlier work about fMRI and basically mapping cognitive function through the brain as you process a task.
Yeah.
How does this overlay that work? Is there a way to look at these areas that enhance or decrease fatigue or enhance function in the time sequence of the actual cognitive process?
Let's see. So there was a study that Dane Cook and Goodrich did,
Yeah.
looking at fatigue and chronic fatigue syndrome, and fMRI, and how the two related to each other. Is that what you're referring to?
Yeah, but I thought they did it again in Gulf War. I think that was a.
Dane is working on that, but in relation to pain, so not fatigue.
Yeah.
And he's on the study. There he is. He's involved. So for what they did before with chronic fatigue, what they did was they said, okay, they give them this other working memory task, which was challenging, and they got the activation from that block, when the subjects were doing that task, and they correlated that with how fatigued they were. So they didn't look at like a progression across time with different, fatigue across time. They just looked at a sort of instantaneous. Here's how you're doing on this task. Here's the brain activation associated with this task, and here's how fatigued you are after you did this task, and how are those two things related? So it was like one part of this.
But I guess what I'm asking is, and I know it's chronic fatigue and not Gulf War, but say you had similar kind of design here.
Yeah.
But you engage in a task, it's fatiguing cognitively, some physiologic things going on. You signal, upregulate a signaling pathway to counter that,
Yep.
which is what you're seeing on your image.
Yep.
And then you complete your task. That was what I would imagine, is that you challenge, react, complete, and that the cognitive speed might be affected by the presence or absence of your ability to do that adaptive thinking.
Right. So that's not a way we've looked at it. So the idea there would be that people with more activation should be responding more quickly. It's just faster reaction time.
Yes.
Right. We could look at that.
That's a thought. And of course, the genomic data that we presented suggests where chronic fatigue and golf were vastly different is in signaling. Is the ability to go around, if you were, a blocked, systemic blocked pathway. So golf 4 is way better at signaling, where chronic fatigue is shutting things off. Golf 4 is turning things on. So there seems to be a very big difference between them in terms of the dynamic modeling to exercise studies that we do in their ability to change course or adapt or change. So I'm just curious if this is a cognitive expression of what we're seeing physiologically in the exercise challenge as a adaptive behavior to counter a less functional space, if you will, for the task.
We can look at this. Yeah.
It's kind of just interesting, because you got the capacity to do that, particularly with your exercise, proposed exercise.
Yeah, we have RB approval for that, so we'll start to know soon.
And we're doing something very similar in women, so we can compare the men
Oh,
to
excellent.
our women data.
Okay.
So that would be
All right.
a little pilot too, it's a pilot.
All right.
Thank you, Dr.
Question after one.
Nope.
When you were talking about this idea of rising to the occasion with some of these tasks, it made me think a lot of intent nurses sort of coming in that way and developing an issue.
Yeah.
And so I wondered, and Dr. Levy talked about the idea of methylphenidate study, and I thought that could be interesting to see if there's something like that happening. You talked about a home-based exercise intervention. Is that trying to get into dopamine somehow, or
No,
what
so
are you trying to do there?
yeah, so we have a methamphetamine study that we're doing in multiple sclerosis to combat fatigue, and that's just started, and so hopefully that will show some efficacy. So a lot, in my work, I've sort of been looking at fatigue more generally, and this is one of the populations that I've been looking at fatigue in, and we've also been looking at fatigue in MS and TBI, like I said. And in MS, there's a guy out in Illinois, Rob Model. His name I have trouble saying properly, but that's the way you say it. He has shown that exercise helps fatigue in MS. He's an MS guy, and so that's why we proposed, or one of the main reasons we're proposing that. And I went to Mike and I asked him if it would work, and Mike said, "Yeah, it should bump up mitochondrial function. It should all work." So I said, "Yeah, okay, let's do that." So yeah, that was sort of independent of the dopamine hypothesis. Yep. A long way to answer your question.
And I've seen some areas where you can see, someone who has a particular neurological issue can perform at the same level as other folks, but they're using more brain areas to actually do that.
Right.
So they're incorporating more areas to perform at the same level. So did you look at concurrent activations in different brain areas?
Not yet. So the analyses we did enables us to look at that. I didn't look at that for this presentation, and I can certainly go back and look at it. In MS, in TBI, that's exactly what you see, and it goes along to what you were saying. It was exactly what you were saying earlier, whereas early in the disease progress, where these guys can still do the task, they're using more real estate to get the task done, more marine real estate, and that's kind of our working hypothesis. It's maybe a little bit of a flat Earth hypothesis. It's like, okay, they're using more real estate, more brain resources to get the job done. That's tiring them out, and it makes sense. And I don't know, it seems a little bit straightforward, but maybe it's true. And whoa, it has been shown in MS. It's been shown in TBI, and we can certainly look at it here. Yeah. All right.
Thank you.
Thank you.
All right.
Well, we're now before lunch. Our last item this morning is a bittersweet one. We are recognizing our retiring RAC committee and staff members, Doctors Bloom, Gollum, White, and staff member Kim Sullivan. And Dr. White, will you begin, or? Thank you.
Floyd, are you on the line?
Floyd.
Floyd Bloom.
Hi.
Hi.
Oh, yes.
Good.
Thank you.
All right. Thank you, Vic. So it's my pleasure and privilege today to recognize 3 RAC members whose time on the committee ends tomorrow. Dr. Beatrice Galome, Dr. Floyd Bloom, and Dr. Kimberly Sullivan. I'm going to show, as I did last time, whoops, how do I make this go forward? The arrow is not working. Do I have to do this other one? Oh, now it's working. All right, well, we know that I can't even run a slideshow, so that probably tells you something about me. But anyway, we're going to start with Dr. Beatrice Galome. Beatrice, you're on the line, right?
Yes, thank you.
Did you get your little certificate plaquey thing? We FedExed it yesterday, no? Okay, it'll come soon. So, but Beatrice should really be called Dr. Galome, and she has both the MD and PhD degrees. She is a true scientist practitioner with a compassionate approach to patients combined with research expertise in many areas that feed her ability to consider the complexity of Gulf War illness. Her emphasis on acetylcholinesterase inhibition as a unifying mechanism for many of the exposures experienced by Gulf War veterans, and her research on mitochondrial dysfunction in Gulf War illness have been critical to the debate on the disorder. Beatrice was the first scientific director of the RAC beginning in 2002. She became chief scientist from 2003 to 2005 and has been a RAC member since then. One of my favorite memories of her service on the committee is her reviews of recent literature on Gulf War illness, in which she would quickly and efficiently go through publications in the field since the last meeting, tying them together and interpreting them with great facility. In fact, the rest of us could hardly keep up. Dr. Galome, we thank you for all of your many years of outstanding, dedicated, compassionate, and thoughtful service to our group, and even more for your past and future contributions to the understanding and care of veterans with Gulf War illness. As I mentioned, we'll be sending you the little acknowledgement. I want everyone to notice that this time, as compared to a year ago, I have more current pictures of people, which has the downside that I can't make as many jokes about them. But I would like to see if anyone else wants to say anything about Beatrice, and if Beatrice wants to say anything. Beatrice?
Yeah, well, actually, I will take the opportunity to mention that my metabolomic partner has sent me an update slide. At least 2/3 of my talk time. I'll mention that his findings distinguishing Gulf War illness and chronic fatigue syndrome include the 2 most affected areas, sphingolipids and phospholipids. In Gulf War illness, 30 sphingolipids were increased, and in chronic fatigue syndrome, 21 were decreased. 6 phospholipids were increased in Gulf War illness and 7 reduced in chronic fatigue syndrome. Branched-chain amino acids were increased in Gulf War illness, reduced in chronic fatigue syndrome, and vitamin C was reduced in Gulf War illness and increased in chronic fatigue syndrome. And vitamin D, cysteine, and GABA and arginine metabolism were also distinguished. And he also did send some nice concrete data and citations on the superiority of metabolomics over genomics in essentially all complex diseases, with genomics being superior so far only in monogenic conditions. But I thought I would just take my time to sort of do a better job
Add to your
completing
time.
that process.
Okay.
And
Thank
also,
you, Beatrice.
How typical.
Go
Thinking
ahead.
the science before herself, huh?
Right, she always does. Yes, go ahead, Beatrice.
I didn't know since my talk only took 2/3 of its time, but was only offered pressing questions. If I could steal my time to answer if there were any additional questions, I would love to.
We're a little short on time, Beatrice, because I have to leave due to a family emergency.
Okay, that's fine.
Okay, so next I would like to acknowledge Dr. Floyd Bloom, who's a distinguished scientist and neuroscientist, who's Professor Emeritus of Molecular and Integrative Neuroscience at Scripps Research Institute in La Jolla. Floyd has a worldwide reputation as a pioneering neuroscientist and neurobiologist. He's been president of the American Association for the Advancement of Science, Society for Neuroscience, and American College for Neuropharmacology, as well as editor-in-chief of the journal Science for 5 years, and an Institute of Medicine member. Dr. Bloom has been a RAC member since 2005, consulting on critical issues surrounding the RAC reports and other scientific issues in Gulf War illness. His support of Gulf War veterans and the mission of the committee has been unwavering, even in times of his own personal illness, so we haven't seen him as much lately as we used to. Having his advice and counsel on the scientific evidence regarding Gulf War illness has been important to both Lee Steele during her tenure as scientific director of the RAC and myself. We've always been able to go to him with questions when we weren't sure we understood things the way they seem to be. Floyd, we thank you for your many years of service and your commitment to the committee and to understanding the issues related to Gulf War illness. We're also sending you a little plaquey thing. I don't know if you got yours either.
Nope, not yet. Thank you.
Okay, so we failed on that one. So do you have anything you want to say, Floyd?
Well, as I wrote you this morning, I'm glad that we established the reality of the Gulf War veterans' illness. I knew nothing about it until Beatrix got me involved locally with putting together a group of pharmacologists experienced in brain acetylcholine mechanisms to see if we could understand the toxicity. And through all these years, when so many people didn't believe that there was such an entity, I think now it's a reality. And my regret is that now that we know that it's a reality, we can't do anything about something that was 25 years ago.
Thank you, Floyd, and thank you so much for sticking to the science.
Absolutely.
Not everyone has done that in this debate, as you well know.
Absolutely.
Dr. Kimberly Sullivan is a neuroscientist and assistant professor at Boston University School of Public Health who began working on Gulf War veterans' health during her doctoral training. Kim has worked for the RAC since 2010 when the contract in support of RAC activities was awarded to me at Boston University. As Associate Scientific Director and Scientific Coordinator for the RAC, Kim is putting countless hours organizing our meetings, talking to speakers and experts in the field, and recruiting scientists with special expertise in neurological, immunological, and other abnormalities that underlie the expression of Gulf War illness in order to produce exciting and inspiring RAC meetings and to help advance the field. She has maintained the website for RAC and supervised all of the logistics of our RAC meetings for the past 5 years. While accomplishing all of this, Dr. Sullivan has developed her own Gulf War illness research portfolio, becoming PI on a DoD CDMRP-sponsored Gulf War Illness Research Consortium, which Dr. Lemon talked about. She has participated as PI or investigator on at least 10 externally sponsored grants on Gulf War illness, and as investigator on more. Her work has supported over $12 million in Gulf War illness research. Thanks, Kim, for all this work and for your research efforts, especially those aimed at understanding the pathobiological basis of the illness and treatment of it. You know better than anyone that the RAC's work for the past several years would not have flourished without you, and I am personally indebted to you for your time and commitment to the RAC. I did bring your certificate, but the frame is in Boston. Because I thought neither of us wanted to carry the frame on the plane. Good thinking. Thank you. Do you want to say anything, Kim?
Sure. I'd just like to say that it's been my great pleasure working on the RAC and dealing with all the veterans I've heard from over the years through the website and phone calls, and I'm more than happy to keep conversing with any of you who have questions. Please feel free to contact me. And also, it's been my real pleasure to actually, when I look back to see how many speakers I had talked to and coordinating meetings and getting them set up, over 100 speakers, believe it or not, in all this time.
I believe it.
It's just been my real pleasure actually interacting with all these speakers, working with them, sometimes bringing them into the field of Gulf War illness research, which has been a tremendous thing to be able to do. And just such an opportunity to call someone and say, we are a federal advisory committee, would you come and talk to us? You're doing something that's similar, and then to get them actually interested enough to submit grants. Just phenomenal. And sometimes to come on the committee, it's just been my absolute great pleasure to be able to be involved in that and to bring those people who are doing fantastic work into this very important field that needs very talented researchers. So it's been my pleasure to do that.
Thank you, Kim.
Don't go too far because we've asked Vic is going to.
All right,
say
and
a little
I
something
have a
for you.
goodbye speech, so
You do?
naturally. It's only 2 minutes long, Jim.
So Vic's going to say something, and then Bob, I'd like to say a few words, and then we'll give you.
I'll get my stopwatch going.
So, Dr. Roberta White has served on the RAC since 2007, served as scientific director since 2008, and during her tenure as scientific director, 2 comprehensive committee reports were prepared, summarizing research across all the multiple disciplines covered in Gulf War illness research. Dr. White is a clinical neuropsychologist and a research scientist. She's professor and chair of the Environmental Health Department at Boston University School of Public Health. She has appointments also in the neurology and behavioral neuroscience programs at the BU School of Medicine. And she was also Associate Dean for Research at the BU School of Public Health until stepping down from that post not too long ago.
May.
And for those of you who aren't aware, Dr. White was one of the very first researchers to study returning Gulf War veterans in the early 90s when studying the Fort Devens cohort of Gulf War veterans in the area near Boston. And she also served as director of the VA Boston Environmental Hazard Center from 1994 to 2005. She also continued to see patients clinically during that time and served as staff psychologist and director of clinical neuropsychology at the Boston VA from 1987 to 2005. Been busy. All this work led to some of the first papers linking environmental exposures from the Gulf War to adverse neurological and other health outcomes in Gulf War veterans. Specifically, she was one of the first to show a relationship between exposures and the changes in the white matter volumes in the brain. The work also produced the first papers linking self-reported chemical and biological weapons and pesticide exposures with chronic health symptoms. More recently, she chaired the integration panel for the DoD Congressionally Directed Medical Research Program's Gulf War Illness Research Program, and continues to serve currently as a member of the Gulf War Illness Research Program. Dr. White has consistently striven to, to understand the neurological consequences of environmental exposures from the Gulf War, and we have no doubt that she will continue to lead the field in this work as she moves on. Thank you, Bobby.
Thank you, Vic.
Bobby, just to follow up on Vic's comments of your vast accomplishments in this field, starting out from the very beginning, actually, one of the very first studies in Gulf War illness and continuing all this time. I thought of a quote that I thought really kind of exemplifies you, and it says, "True leaders don't create followers, they create new leaders." And I think that you've led this field from day one, and you have really created new leaders, whether it was in your capacity on the RAC, teaching other investigators how to do this research and how to lead in it, and also on the integration panel at work as well, doing the same. So as a true leader who has dedicated so much time and effort to this field to help veterans, because we know that you have a strong feeling for Gulf War veterans and always have, that we actually would like to give you this clock.
Thank you.
Mind your dedication.
Travel from Boston and is going home.
Just make sure I have the box, or I'll destroy it on the
Yeah,
way
I was just gonna say,
home.
make sure you have the nice box if you can.
Oh, thank you. What?
You can
She's gonna
FedEx
take a
it.
picture.
That's nice.
Thank you, Kim. Thanks so much. When Dr. Fran Murphy called from the VACO Office of Public Health 22 years ago to ask me for help, and also that of Jessica Wolf and other investigators at the VA on the newly emerging health problems of Gulf War veterans, I had no idea that our work with the Fort Devens cohort would lead to the depth and breadth of research and personal involvement that would ensue. I knew that the answers to the questions might take a long time because the Agent Orange story was close and familiar, and that took over 20 years also. But I had no idea that I would work this intensely on this issue for so many years and in so many different ways. The 25th anniversary of hostilities is rapidly approaching, and we know a great deal about the illness. Promising treatment approaches are in the preclinical and clinical stages of testing, and there is hope for improvement in the lives of Gulf War veterans. It seems so late. For that, I feel so sorry. Despite my wishes that our progress down this path had been faster, I have found these years to be extremely compelling and rewarding. The scientific work has been exciting. Interactions with the VA and other government agencies have been intriguing. Dealing with the media has been both fun and challenging. Working with Congress, Institute of Medicine, and other advisory bodies has been fascinating, and I have loved working with CDMRP on their research program. Most compelling has been working with Gulf War veterans as patients and colleagues. Veterans' contributions to research review and to the design and implementation of scientific work have been invaluable. Their advocacy for policies to support ill veterans has been inspiring, and I admire their accomplishments greatly. Jim Binns and Lee Steele spent a lot of time convincing me to work with RAC and to take on the scientific directorship. I was really hesitant because of everything else I had to do. There have been times when the challenges have seemed overwhelming, but I have had a particularly gratifying life and professional experience in doing this work, for which I'm very grateful. It's something I would never undo. VA has funded the RAC through a contract to my university, which made it much easier for me to manage the associated expenses and tasks, and I've developed new colleagueships and enjoyed working with many VA colleagues. Thanks to Vic Kalasinski, Tim O'Leary, and others who have made this possible. Also to Bob Yeager, who seems to be my personal itinerant caterer and who often provides me with food when I come down here to do stuff, and even some hugs. To former and current RAC members, I thank you for the rich contributions to the work of the committee. It is unique in my experience to participate in committee meetings that have special implications for the lives of individuals and at the same time are scientifically exciting. This is due to your contributions. Special thanks go to Jim and Lee, Jim O'Callahan, Fiona, Nancy Klimas, Bill Meggs, Joel Graves, Marguerite Knox, and Anthony Hardy, as well as Kim, Beatrice, and Floyd. You have all gone out of your ways to help when we have needed you. Most of all, I wish to thank the veterans who faithfully attend our RAC meetings. You are our inspiration, and your support and contributions have made all of the difference in this work. I have gotten to know some of you so well that I recognize your voices on the phone. Denise or Angie or other people can call me, and if they don't say who they are, I know who it is. Sometimes they forget to say. I hope that you will all stay in touch. Thanks and ever onward to Steve, Fran, Jim, Fiona, and Nancy. I look forward to watching your progress, and thanks to everyone for my time here.
Well, maybe I will say a word to my fellow remaining members who will be here at our next meeting. I personally feel a little bit like the San Francisco 49ers watching Jim Harbaugh walk out the door and wondering how we will continue. Beatrice is our link to the beginning of RAC and our last link to the beginning of the RAC, despite your youth. And Floyd is, in my world, in the world of science, a giant figure, truly a giant figure in modern biomedical science. And Dr. Bloom's presence cannot be replicated. And Dr. White, in addition to her own work, which has been so important, has been the intellectual glue for our commission for almost a decade. And Kim has made everything we've done work on autopilot, it seems, and we can always be confident that the meetings and the organization will be perfect because she knows every corner of the field. So to those who will stay on for the 2016 year, and for the new members who will repopulate, I think we are standing on the accomplishments of giant individuals, and I know that I'm speaking for all of us, that we will do our best to do as well as we possibly can to keep them going. So thank you all again. It is exactly